This is Mr. Russell, your AP biology teacher, and we're going to be looking today at our first section of notes and our first PowerPoint for looking at how life began on Earth according to science. Well, according to science, there were 4 things necessary for life to emerge. The first was an abiotic synthesis of amino acids and nucleic acids. If you go back to our early days of biology, you'll remember that bio, meaning life, so biotic is living organisms, and A being against or non, abiotic being non-living organisms. Well, the RNA molecule is believed to have evolved first. It's not as molecularly stable as DNA. If you remember that nucleic acids such as RNA and DNA, RNA being ribonucleic acid and DNA being deoxyribonucleic acid, are essential for storing, retrieving, conveying inheritance through molecular or other molecular information on constructing components of living cells. So this just gets passed down from one generation to the next. The third thing for when we're talking about this abiotic synthesis is being able to make amino acids. Amino acids are the building blocks for proteins, and proteins are often referred to as the workhorse of the cell. So the majority of a cell or organism in biomass or its dry weight is mostly proteins. Well, and here you can see proteins in their natural form. If you remember, we talked about early on, and we're going to spend a lot of time talking about the functions of protein, their structure. Here you're just looking at their natural form because they function according to how they fold. We'll get into that one later. Now, second thing that's necessary for life to have begun on Earth according to science is that monomers must be able to join together to form complex polymers using energy that is obtained from the surrounding environment. Now, if you think about the term monomers and polymers. Mono, what do you remember? What does mono mean? I think of it as being one or single. So if we think about single units, monomers, think of this as like Legos. If I take one Lego, a monomer, attach it to another one, and then to another one, and eventually I can build something like a house, or a street, or a car, or maybe even an airplane. And you have what's called a more complex structure, which would be a polymer. Poly meaning many units. So monomers build polymers. One Lego put together with many more Legos, you can build your own little Lego house to live in. Well, oftentimes this can be seen in monosaccharides. There's that term again, mono. Saccharides being sugars, so single sugars or simple sugars make polysaccharides. This is for energy storage or cell walls in plants. So simple sugars build more complex sugars. It's made in, if you go back to, you remember our phospholipids in our cell membrane. This is another example of monomers building polymers. It's also seen when we're talking about being able to making the messenger RNA used in making proteins using amino acids. We'll go into this a lot more later. I know that you're not going to remember all of this, but at the same time, these are things that you've got to be looking at and studying. So we're using mRNA or messenger RNA making proteins to be able to make amino acids, or using amino acids. Now, it's also seen in making those chromosomes out of strings of DNA molecule. So, if you remember, we go back and we look at RNA and DNA, which brings us to the third step necessary for creating life on Earth according to science. RNA and DNA molecules form and gain the ability to reproduce and stabilize by using chemical bonds and complementary bonding. If you remember back to your early intro biology class, you'll remember that there were nitrogenous base pairs, and those base pairs have to bond together. Adenine always goes with thymine, AT. They always go together. Guanine always goes with cytosine, GC. That's when we're talking about DNA. The only exception to that is that when we're talking about DNA, adenine would then pair with uracil rather than thymine. And we'll get into that one a little bit later as well. So, we look at the single strands of DNA and RNA molecules. They're also held together by the covalent bonds. And in the picture that I'm showing you here, we're looking at the blue. If you think of this as a spiral staircase, the handrails, or what's holding the steps together, the blue part is going to be held together by your covalent bonds. Maybe you can see it better here. The orange strands are held together by covalent bonds, and then the blue strands themselves are held together by covalent bonds. Very strong bonds. Now, these are ionic bonds as well. So, when we look at the A and T, how they're held together, and the G and C, or the A and the U, so blue to blue, or orange to blue, then we're talking about hydrogen bonds. We'll discuss this one later in the unit, but these are considered weak bonds. If you think about, I think of hydrogen bonds being kind of like high school dating. Sometimes people are able to stay together for a while, then all of a sudden they break up, and then they get back together and they break up. Whereas a covalent bond should be a much stronger, longer lasting bond, more like a marriage. Well, when we're doing this, we're talking about what's going to happen if we're building these things. So we're saying there's 4 steps. The first step was the abiotic synthesis of amino acids and nucleic acids. We said the second step was monomers joining together to be able to build more complex polymers. And then RNA and DNA molecules will be the third step. These form and gain the ability to reproduce and stabilize by using chemical bonds and complementary bonding. Then the fourth part would be the evolution of a protobiont. Protobiont membrane is just the first life form. This is made up of phospholipids and proteins that will help to keep the cell intact. Okay, so when we look at how do we know this? This is, remember, according to science. In order for scientists to be able to make a statement, they need to be able to conduct experiments. They need to be able to reproduce or produce evidence in a reproducible experiment. Well, so we're going to look at the evidence that's going to support each of these steps that we've just been talking about. Well, remember, step 1 was the abiotic synthesis of amino acids. Well, Stanley Miller and Harold Urey, they conducted an experiment in 1953 that supports this claim. This experiment took the inorganic gases hypothesized to have been in early Earth. We're talking about hydrogen, methane, ammonia, and water vapor, and formed a variety of organic molecules from these. And we're talking about they were able to form things like amino acids and oils. These organic molecules are the building blocks of life forms according to science. So the energy source for the formation was an electrical spark that was to represent lightning in the early atmosphere. The gases hypothesized to be present in the early Earth's atmosphere came from data associated with analyzing active volcano eruptions. And we talk about how we look at volcanoes would have been very active and present in the early time period. As well, there's still today the big ring of fire, these volcanic islands. Hawaii is still being formed, it's growing all the time. So these organic molecules would have accumulated in the Earth's early oceans over millions of years, thus making it possible the formation of more complex structures, as we saw in step 2, which is forming monomers, taking monomers and forming polymers. Now, we're talking about this, it's saying researchers have shown that by exposing pyrite, which is just fool's gold, also exposing sand or clay to the intense heat of sunlight, and in the presence of tidal action of the ocean, that complex polymers can form from simple building blocks of amino acids and oils. Remember step 1. So these happen and they get dissolved in water, and this process takes place. This complex polymers would then also have accumulated in the Earth's oceans over millions of years, and then making possible the formation of more complex structures in step 3. Remember, step 3 was the RNA formation and reproduction. So the experiments of Thomas Keck, I believe is how you say his name, hopefully I didn't mess that up, and Sidney Altman, they've shown that small strands of RNA molecules can act as enzymes, and enzymes are just biological catalysts for chemical reactions. Think of enzymes as speeding up and slowing down reactions, kind of like the school bell. When you hear the school bell, that lets you know that you either need to get to class pretty quickly, or lets you know that class is over. So it changes, it speeds up and slows down reactions. So the enzymatic RNA molecule, these are called ribozymes. These are small strands of RNA that would have been the product of step 2 that we talked about earlier. And RNAs are great for being enzymatic as well as temporary. It stores and transmits molecular information as seen in protein synthesis using messenger RNA. So if you think about it, storing, it's kind of like the bank. I think of DNA being kind of like a bank where it holds your money all the time, and you go and you get money out of the bank. That would be like RNA. It's very temporary. You're going to spend that really quick. You're going to take it from one place to another. So according to science, over millions of years, a more stable and longer lasting molecule would have evolved. That molecule is going to be DNA. It would be the more stable, longer lasting means for strong molecular information and transmitting this important information from a cellular generation to the next in the process of binary fission or even in mitosis. So this would create the ability to reproduce cells or organisms which would come into existence within step 4. If you remember in step 4 we talked about earlier was the formation of a protobiont membrane. So experiments have shown that lipids, remember phospholipids, so we're talking about lipids and other organic molecules. They form in step 1, 2, and 3. They can form membrane-bound structures similar to a cell membrane. This formation allows the complex of molecular interactions to occur in a safe inner environment away from the outside environment. In simple terms, you have an inside and an area separated from an outside area by a lipid or oil-based structure referred to as a membrane. So over a long period of time, it's thought that these enclosed environments would have become the first prokaryotic cells. These prokaryotic cells would possess DNA and RNA molecules inside them. So the RNA would be used to construct ribosomes, which are needed to link amino acids together to be able to make proteins. So the DNA would be for storing, excuse me. So the DNA would be for storing that protein-making information as well as passing it on to the next generation. Therefore, according to science, life could have came into existence over a very, very long period of time. All right, so this is your first day's worth of notes. You may want to go back and look through it, look through some videos online, study these, and we will see you on the next round.